Hello class, this is chapter 4. It'll be just part 1, which is the cellular level organization. It will introduce us into microscopes, and the next part will be 2 and 3. So it's important to know for this chapter that cells are a group of organelles which are made up of molecules which are made up of atoms. And then if you add cells together we get tissues. If we add tissues together we get organs. So along that hierarchy, cells are the smallest thing that is considered alive. Cell theory states that. It also states that all things that are alive are composed of one or more cells. Cells are the basic units that make up organisms. We are going to have to compare animal cells to plant cells, to also prokaryotic cells like bacteria. They are slightly different in structure, but they generally have shared parts. We need microscopes to look at cells, which is one reason I found them so interesting as a kid. They were alive, but you couldn't really see them without help. You need to know sort of the range for seeing stuff, and so this range up here is for the visible eye. And you can see organisms, you can see something like a frog egg with the visible eye, or a fish egg. You can also see a human egg. It would only look like, it's only about the size of the tip of a pencil, so it would only look like a dot, but you can see a human egg with the naked eye. This next bar is going to be for light microscopes. Light microscopes use light to illuminate their specimen. And typically the light has to be able to pass through the specimen. You can use it to look at larger cells like eggs, but even smaller cells like animal and plant cells and bacteria. Light microscopes don't give you a ton of detail, but they do let you look at the tissue hierarchy and the cell hierarchy. Some organelles you can see, like chloroplasts and nuclei, but other little bits are hard to identify that are in the cell when you're looking at them with a light microscope. So this third bar here is, so this is the first bar. It's with your eye. This is the second bar. I really miss the pointer. That's the second bar. That's the light microscope. And then the third bar is going to be the electron microscope. And the electron microscope uses an electron beam instead of a light beam to look at your specimen. Basically, electrons are going to be blocked, or they're going to change direction when they hit solid structures, and then they're going to pass straight through when there's less solid structures. Excuse me. So you can look at cells with it and look at cells in high detail, high magnification. You can even look at smaller structures like viruses and proteins. We can detect atoms, but we can't really see atoms. So that's why the arrow stops just before the atom. We can see molecules if they're large enough. So this is a picture of a molecule, and it is within the range of the electron microscope. The light microscope, the max magnification you can get with it is typically around 1000x total magnification. When I was using the electron microscope, I was in the magnitude of tens of thousands to 100,000 times. So 100,000, 200,000, 500,000. That was sort of the limit I typically got to with electron microscopes, but nowadays it may be more powerful. Cell sizes are typically in the micrometer range, although some really large ones can be in the millimeter range, like the onion epidermis, if we take a look at that this semester. The reason they're this small is so they can have adequate surface area to volume ratio. And that's so they can exchange materials through using diffusion as the driving force. Diffusion happens pretty quickly, but its range is pretty short. So you have to have lots of surface area in order to have efficient exchange with diffusion. So in smaller cells you can do diffusion really easy. Larger cells, the way they get around it, they compartmentalize. So you're having diffusion between compartments, and each compartment is small. So not only will they have lots of surface area, but they'll have lots of internal surface area as well. So here, just doing math, this represents a large cell, this represents a bunch of small cells. And then you can see the surface area to volume ratio with each scenario. And you can see that as those cubes get smaller, you have a higher surface area to volume. So this is why cells are small, so they have high surface area to volume relationship. So they can exchange toxins and nutrients with diffusion. And that would be a common theme when you get to anatomy and physiology, structures that increase the surface area. Alrighty, so let's talk about a compound light microscope. If you're doing online labs, you're simulating these a lot, but in lab, we use these a lot. And again, we use light to eliminate the specimen. The light is focused by lenses, and then the image is bounced around by mirrors to get to some oculars. You know, nowadays we rarely use the oculars with the camera. Typically in the body tube is a camera, and that camera is hooked up to a computer. But in our labs, we typically do use the oculars, and so we use a lot of mirrors to get the image to the oculars. Again, the max magnification is around 1,000 times. and we're limited. We could go to higher magnifications, but we can't get better resolution. So our limit typically is 1000x, and there's a resolution limit as well. So this is the type of image you would expect to see with a light microscope. Here's the mirrors I was talking about. Here are your oculars. They let you view your specimen. Your specimen will be on a stage. That stage will have a hole in it where light can pass through, so your light's passing through this way, bouncy, bouncy, bounce, and then up to your eye. And right here are your objectives. They will magnify your specimen at different levels. And you can focus your image with the focus adjustment wheels. A lot of microscopy today uses electron microscopes. You need to know the difference between transmission and scanning. Transmission electron microscopes let you look through a cell, so the inside of a cell. In high detail, and so you could look at organelles and sometimes molecules like proteins inside the cell with a transmission electron microscope. With the light microscope, you can see cell shapes and larger organelles, but again, while you can see smaller organelles, you can't see it with enough detail to tell what it is. So typically you can tell chloroplasts because they're green, and you can tell the nucleus because it's large and central and round. But that's usually all you can really tell. When you're using a light microscope, with the transmission electron microscope, you can usually identify every single organelle that I will talk about later. So yeah, that's transmission. So here's an example of a transmission electron microscope, and again you can see a bunch of different organelles. In the cell, and hopefully we'll get to look at pictures a little bit closer up later. So a scanning electron microscope also has this high resolution power, it has the high magnification power, but typically we're looking at the structure of a cell, so the surface. And its images are in 3 dimensions. And you can pseudo color those images, and so when you see like the compound bug eye, that is typically with a scanning electron microscope. So scanning electron microscope is used for cell surfaces, and you can see larger things in extreme detail, like bug eyes and things like that. And so here you can see the 3D image. This is a parasite. Oh, sorry, it's not a parasite, it's Euglena. Anyway, you can see it's flagella, and it's a 3D image, it's not a flat image. With the transmission and light microscopes, we typically get flat images, although there are some technologies now where we can create a 3D image from lots of data. But it's not a true 3D image like the scanning electron microscope is. The problems with microscopes are magnification, resolution, and contrast. Most cells are clear, so it's really hard to tell a cell from its background, and that is contrast. So often we will stain our cells so we can see them better, and that gives us more contrast. There are also specialized microscopy techniques that help with contrast. Resolution is the ability to tell that 2 points are separate. So if you put 2 points really close together, sometimes it's hard to tell that they're two separate structures. And the smallest distance that you can tell two dots are separate is the resolution. And the resolution is really what limits the light microscopes. The resolution is much more finer with electron microscopes. And then we talked about magnification already. And just like there's special microscope techniques and microscopes for contrast, there's also techniques and microscopes for resolution. Confocal is one of the ways to get better resolution. It's a different type of microscope. I only got to use those a couple times, but the images are pretty. And a lot of microscopy now uses a camera. So here you can see a light microscope image. Again, you can see the general structure of the cell. You can see some bits on the inside, but it's hard to identify most of them. So light microscope is useful for identifying the type of cell, but it's usually not good for studying specific structures without specialized techniques or microscopes. Here is an image from a transmission electron microscope. Not only can you see the shape of the cell, but you can see all the internal bits at much higher resolution. So you can usually determine what they are, like Golgi or mitochondria or nucleus. The problem with transmission electron microscopy is that these things have to be dead and they have to be extensively processed. So some of the things you're seeing could be artifacts, but most of the time it's not. And this last image over here is a scanning electron micrograph. And remember, it's the cell surface and you get 3D images. So I know the difference between these three, sort of strengths and weaknesses, what they're used for, and you should be good for the exam on this part. Here they're just showing you different techniques to see a cheek cell. Here's one that's stained, and here's one that's using one type of contrast technique, so it's not stained, it's alive. Staining does typically make the cells sick or dead. And then here's another type of contrast enhancement. With DIC, you can do it with most microscopes. With phase contrast, you need a special microscope. So that's it for this part. The big takeaways from this should be the three main microscope types and being able to tell the difference between them and their function and what they're good for. And then also the concept of cells, so cell theory, and the surface area to volume importance. Why are cells small?